In 1918, an influenza virus killed between 50 and 100 million people, more than the entire First World War, in under 2 years. We call it the Spanish flu, and for decades, scientists have asked the same question: could it happen again? Right now, in dairy farms across the United States, in poultry farms across Southeast Asia, in wild bird populations spanning every continent on Earth, a virus is doing something that has researchers at the WHO, the CDC, and institutions across the world watching more closely than almost any other pathogen on the planet. It is mutating, and it is learning how to infect mammals. Today, BodyCode goes inside the biology of H5N1 avian influenza. Not the headline version, the molecular science of what is actually happening, what the mutations mean, why dairy cattle changed the risk calculation, and what the honest scientific assessment of the threat actually is. Because this is not panic. This is the science you need to understand before it becomes impossible to ignore. To understand why H5N1 is different from every other flu strain, we have to understand the receptor problem. All influenza viruses enter human cells the same way. They use a surface protein called hemagglutinin to bind to receptors on the cells lining your respiratory tract. Here is the critical biology. Human respiratory tracts have 2 types of receptors. Alpha-2,6 sialic acid receptors line the upper respiratory tract, your nose, throat, and upper airways. Alpha-2,3 sialic acid receptors line the deep lung tissue. Seasonal human influenza viruses prefer alpha-2,6 receptors. This is why they spread so efficiently. You breathe in the virus. It binds immediately in your upper airways. You sneeze. Someone else breathes it in. The chain continues. H5N1 avian influenza prefers alpha-2,3 receptors, the deep lung receptors. This is simultaneously the reason it does not yet spread efficiently between humans, and the reason it is so deadly when it does infect someone. When H5N1 reaches a human, it travels past the upper airways and lodges deep in the lung tissue. The immune response to deep lung infection is catastrophic. Cytokine storm, fluid flooding the alveoli, the same drowning mechanism we covered in the hantavirus video, but faster and more aggressive. Since 2003, the WHO has confirmed 993 human cases of H5N1. 477 of those people died. That is a case fatality rate of 48%. For comparison, the 1918 Spanish flu had a case fatality rate of approximately 2%, and it killed up to 100 million people. H5N1 is 24 times more lethal. The only reason it has not caused a pandemic is that single receptor preference. It cannot yet spread efficiently from one human to another. The word "yet" is doing enormous scientific work in that sentence. Now, the honest question: should you be panicking right now? No. And here is the science behind that answer. The specific genetic leap that H5N1 needs to become a pandemic virus requires acquiring efficient human upper respiratory tract binding, the alpha-2,6 receptor preference, without losing its replication fitness. This is not easy. Influenza viruses that gain one advantage often lose another. Laboratory research published in the CDC's Emerging Infectious Diseases Journal in May 2026 found that contemporary H5N1 isolates still replicate poorly in human nasal epithelium compared to seasonal flu strains. The upper respiratory tract barrier is still holding. Furthermore, the global pandemic preparedness infrastructure is in a fundamentally different position than it was in 2019. mRNA vaccine technology, developed and validated during COVID-19, can produce a targeted H5N1 vaccine candidate in weeks once a pandemic strain is identified. Pre-positioned stockpiles of H5N1 vaccine candidates already exist in multiple countries. The surveillance systems that missed the early weeks of COVID have been substantially upgraded. WHO's Global Alert Network is actively monitoring every H5 detection in real time. This is not 2019. The world is watching, and it has better tools than it has ever had. In 2024, something happened that changed the risk calculation. H5N1 was detected in dairy cattle herds across the United States. This was not supposed to happen. Cattle are not birds. H5N1 had never been documented to establish itself in cattle populations before. And yet, by mid-2024, it had spread to dairy herds across multiple states, and dairy workers began testing positive. This matters for one specific biological reason. Every time H5N1 infects a new mammalian host, it has an opportunity to acquire mutations. Mutations that make it better at surviving inside a mammalian body, better at binding to mammalian receptors, better at replicating at mammalian body temperatures. Scientists monitor specific mutations in the virus's polymerase gene, particularly in a region called PB2. Two mutations in this region, designated E627K and D701N, are known to dramatically enhance how efficiently influenza viruses replicate inside mammals. These mutations have already been detected in fox isolates, in mink isolates, in some cattle-linked sequences. Published research from February 2026 documented that clade 2.3.4.4b H5N1, the currently dominant global strain, has expanded into an increasing range of mammals, and that sustained transmission has occurred in both minks and dairy cattle. This is the mechanism of pandemic emergence, not a sudden event. A gradual accumulation of mutations in a virus that is finding new hosts, learning new bodies, and getting better at surviving in them. In November 2025, the United States confirmed a human case that had never been seen before in the entire history of influenza surveillance. A person infected with Influenza A H5N5. Not H5N1, not H5N6, H5N5. This was the first human infection with this specific subtype ever recorded globally. Contact tracing found no further human cases. No human-to-human transmission was detected. But here is what that case tells us. The H5 family of avian influenza viruses is not one virus. It is a constantly diversifying family of variants, each with slightly different surface proteins, slightly different receptor preferences, slightly different replication characteristics. And as that family expands, the probability that one of its members will acquire the specific combination of mutations needed for efficient human transmission increases. This is not speculation. This is elementary probability applied to viral evolution. In the first quarter of 2026 alone, WHO tracked 13 avian and swine influenza infections in humans across multiple countries, including a fatal H5N1 case in a child in Bangladesh, H9N2 cases in China and Italy, and the continued expansion of H5 subtypes into new geographic territories and new host species. The surveillance data is not showing a stable situation. It is showing a system under increasing pressure. So what does the scientific community actually say about the probability? The honest answer is that nobody knows, and any source telling you they do is overstating their certainty. What researchers do say is this: the conditions for pandemic emergence are more present right now than they have been at almost any point in the documented history of H5N1, a globally dominant clade, expanding mammalian host range, detected mammalian adaptation mutations, an unprecedented novel subtype human infection, and continuous viral replication across multiple species, each replication cycle another opportunity for a critical mutation. The gap between the current situation and a pandemic scenario is not impossibly large. It requires a specific combination of mutations, but it does not require a biological miracle. What the responsible scientific position says is this: we are not in a pandemic. We may never be in an H5N1 pandemic, but the probability is not negligible. And the appropriate response to a non-negligible probability of a catastrophic event is not panic, it is preparation. At the individual level, preparation means exactly what it has always meant: avoiding direct contact with sick or dead birds, washing hands thoroughly after any animal contact, seeking medical attention promptly for respiratory illness with fever, especially after any potential animal exposure, and telling your doctor if you have had contact with poultry or cattle. These are not dramatic actions, but they are the ones that matter. In 1918, nobody understood what was happening until it was already everywhere. The science did not exist. The surveillance did not exist. The tools did not exist. In 2026, we have all three. We know what H5N1 is. We know what mutations to watch for. We know where the virus is circulating and how it is evolving. We have vaccine platforms ready to deploy. We have the institutional memory of COVID-19 with all its failures and all its lessons. The gap between knowledge and action is the only gap that matters now. And that gap is narrower than it has ever been. Understanding what H5N1 is, the receptor biology, the mammalian adaptation pathway, the honest risk assessment, is not just scientific literacy. It is the difference between responding rationally to a real and documented threat and either dismissing it entirely or surrendering to panic. Neither extreme serves you. The biology does. This is what BodyCode exists for: not to frighten you, not to dismiss real science with false reassurance, but to give you the actual mechanism, the molecular reality behind the headlines, so that you can think clearly about your own biology and the world it exists in. If this video gave you something that no headline did, share it with someone who needs it. And if you want to keep decoding the systems that run your body and shape the world around it, subscribe. We will see you in the next one.